Hey everyone, remember last time we were talking about the idea of polymers, which was a long chain molecule, and monomers, which was a small unit that combined in a repetitive manner to make the polymer. Now we knew that condensation polymers were made by the elimination of a small molecule like HCl or water from the functional groups of the monomers. Monomer had the functional group like carboxylic acid, the hydroxyl, or the amine functional group. And monomers needed these functional groups on both sides. Like last time, we used a monomer with carboxylic acid and the alcoholic group, and we made ester polymer out of those. In today's video, we're going to see a new kind of functional group attached on the monomer, which is the nitrogen and two hydrogens. It is called amine functional group. If your carbon chain has a nitrogen with two hydrogens on it, it's called amine functional group, exactly like carboxylic acid is a functional group or hydroxyl is a functional group. Amine is also a functional group. This molecule has two functional groups of the amine, so I'm going to call it diamine because it has two amine functional groups. It has two NH2 groups. So this is a diamine over here, and it's going to be a monomer for our new polymer. On the other side, I have a dicarboxylic acid because it got two carboxylic acid functional groups on both sides of a molecule. What is this hexagon and the circle? We don't care about it. We don't have to worry about it. We just know that there is carboxylic acid functional group on both sides, on left and right. Now, the amine always loses the hydrogen atom, and carboxylic acid in our case is going to lose the OH atom. Because amine had a hydrogen atom to lose and carboxylic had the OH. So you copy the whole structure with the nitrogens and one hydrogen because amine has lost one hydrogen from both sides. You copy the other monomer, but this time you don't copy the OH groups. So you copy the hexagon, the ring, the carbon double bonded oxygen, but you don't copy the OHs on both sides. Now you connect both the monomers and you make extended bonds from left to right. This is your polymer ready. The amine lost the hydrogen from both sides, the carboxylic acid lost the OH. What you have in the middle is amide linkage, nitrogen, hydrogen and carbon double bonded oxygen. Similarly over here you got two different monomers. The blue one has amine on the left side of the molecule, which is NH2 functional group, and it got a carboxylic acid on the right side of the molecule. We don't worry about the fact that whether it's on the left side or the right side. Similarly, the yellow monomer also has amine on the left side and carboxylic acid on the right side. Technically, these molecules are amino acids because they got amine and acidic functional group. The amine always loses the H and the carboxylic acid always loses the OH. And this way you get water molecules out of it. You start redrawing the structure from the nitrogen along with the hydrogen on the top of it. You draw this box which could be anything at all. You draw the carbon double bonded oxygen and you draw the line because the OHs are gone, remember? Then you start copying from the other nitrogen. You copy the nitrogen, you make the hydrogen on top of it, you make another box which is going to be the yellow box, the carbon double bonded oxygen, and you make a line on the right. Now you know that this is your polymer because it's going to be a continuation. This is the blue monomer which I'm shading with a blue pen. This is the yellow monomer over here, and now you connect them. I forgot to connect them actually. The line should have been a little longer. So carbon double bonded oxygen and NH2 are connected. This part is your main linkage. It is known as amide linkage, AMIDE, amide linkage. It is very similar to the idea of ester linkage, but it's a new linkage. Now, since we are done, let's try the concept of hydrolysis. You know, hydrolysis is the opposite of polymerization. So it's the breakdown of a long polymer into its monomers. It is the opposite of polymerization. Here, it's a breakdown of a long polymer into its monomers. You're breaking it down. For example, the word hydrolysis is a combination of hydro, which means water, and lysis, which means break down. So over here, every time you will have your polymer with whatever linkages. For example, here I can see the ester linkages right in the middle. This is a blue monomer, there's a pink monomer, and there's an ester linkage in the middle. So you break your main linkages down, and then you identify your monomers and you separate them. So it is breaking the polymer down into the monomers which were used to make it. So it's the opposite of polymerization. For example, here you can see a polymer. You can see the ester linkages right in the middle. It's a carbon double bonded oxygen, and there's another oxygen right beside it, so it's none other than the ester linkage. If you noticed really quickly, focused really well, I should say, you can identify there's a blue monomer, which is the oxygen, then carbon and carbon double bonded oxygen, and then there's this other monomer, which is the green one. It is a repeated monomer coming again and again, joined by the ester linkage. So you break the ester linkage down. After breaking it, copy everything else. Break your monomer exactly the way it is. Don't add or subtract anything at all. Draw the carbon double bonded oxygen. On the other side of the sheet, draw the other monomer, which is exactly the same one, but let's redraw it. The carbon with 3 hydrogens, the other carbon with a carbon double bonded oxygen, redraw it. Now you have to add H's and OH's on the monomers to complete it, because this is monomer but not complete. So you add H's along with the oxygen and OH's along with the carbon double bonded oxygen, so that you get OH on the left side and carboxylic acid group on the right side. Let's try this one. Again, you can see there's an ester linkage in the middle. This purple thingy it could be a carbon chain, it could be a hydrogen atom, it could be any part of the carbon chain molecule. We don't care about it. Similarly, the pink circle could be anything at all. It could be a very long chain, a very short chain. For all we know, it could be anything at all. So it could be whatever. But what we focus on is the ester linkage right in the middle. You break it down because that's the main linkage. You separate the carbon double bonded oxygen with the other oxygen. You copy everything down without adding a single atom right now. So carbon double bonded oxygen over here, the purple box, the oxygen on the left. This is the purple box being done. On the other side of the sheet, with a proper gap so you don't get confused, draw the oxygen, the pink circle, the other carbon double bonded oxygen, the line. Do it. Once you are done redrawing these monomer parts, you have got to complete it by adding H's on the oxygen atom and OH's on the carboxylic acid group. Left gets a hydrogen atom, the right side gets a carboxylic acid group. So that's how it's completed. This is one monomer. The purple boxy thing is monomer number 1, and the other one is a monomer number 2 over here. You can see you have hydroxyl group on the left side of both monomers, the purple box and the pink circle. Similarly, you have got carboxylic acid functional group on the right side of both monomers. You can't leave the carbon double bonded oxygen hanging in there. You have got to draw OH around the carbon double bonded oxygen so it becomes a complete group, and the left side also get a simple hydrogen so it properly becomes a hydroxyl group. I hope the idea of hydrolysis is really clear to us, so in the next video we could be trying more and more past papers for it. Thank you guys, stay tuned.